So, as we know, psychedelic plants have a long tradition of use in shamanism and indigenous use, going back at least 60,000 years, probably much older than that. And that's really what we have information. That's the antiquity that we have evidence for from rock paintings and that sort of thing. So they show a long alliance with humanity, and around the shamanic use of entheogenic plants and fungi have developed archaic techniques of ecstasy, ways to essentially soul travel, to understand, to explore other dimensions of consciousness. Where those dimensions are is a matter of debate, even 60,000 years into it. Does it come, is it merely something produced by the brain? Are these truly other dimensions or not? And we really don't know. And despite our scientific sophistication, we really can't say. But we know that it's endlessly fascinating. And, oops, sorry. And in a lot of cases, there's a great deal of sort of gnosis put on the plants. We have these notions that plants themselves are gods, they're intelligences, they're deities in some ways, or they're at least treated that way. And part of shamanism is that these are the specialists in the archaic techniques of ecstasy. These are the folks, the men and the women, who undergo special training or have initiatory experiences to learn how to navigate in these transdimensional realms. And they use these plants to communicate with what are understood in these indigenous societies as gods or spirits. Healing is an integral part of shamanic practice, and they get their knowledge of plants and healing modalities through their transcendental navigations in these realms. And the shaman is a very sort of double-edged, two-faced figure in a sense, I mean, ambiguous. However, he or she fills a very important function in the society. He can locate where the game is going to be. He does important functions for the sort of integration of a society, an indigenous society, into its ecology. So we have that much data. We know, probably most of us from our own experience, and certainly from the literature, that psychedelics are notorious even for their changes in visual perception and other perceptual modalities that they can elicit. For example, visual hallucinations and distortions of the visual apprehension. And you see these visual effects of psychedelics reflected again and again in many cultures, in the iconography of indigenous cultures, such as this contemporary painting by Robert Venosa on my left, the Huichol yarn paintings, and also at the lower part, the Huichol jaguar mask. These are basically information that are derived from these shamanic experiences. And when it comes to pharmacology and trying to understand the nuts and bolts of all this and how does this work, we know that the neurotransmitter serotonin, which is itself a tryptamine, like many of the entheogens or the psychedelics, it is 5-hydroxytryptamine. That's where the term 5-HT comes from. It's a master neurotransmitter in the brain and also has peripheral effects as a kind of hormone-like compound. And psychedelics interact more or less selectively. There are many subpopulations of serotonin receptors. About 14 different subtypes have been identified and characterized. The gene sequences have been articulated, and we know a lot about the serotonin receptors. And the two that emerge as most relevant to the action of psychedelics are the so-called 1A, the serotonin or 5-HT1A and 2A receptors. Other psychedelics, other psychoactive drugs may resemble psychedelics in some respects experientially, but they're not true psychedelics, I like to say, if they don't conform to this 1A, 2A pharmacology modality. Here's a nice picture of LSD nesting very nicely in the serotonin 2A receptor. This is a computer-generated graphic. This is part of the work done by one of our Hefter members, Dr. Nichols, has spent a lot of time trying to characterize the nature of the binding, the nesting site, if you will, how these psychedelics fit into the 2A and 1A and other receptors and do what they do. And a nice side effect of that is that in order to study this binding process, he's had to make a great many analogs of psychedelics, of the natural psychedelics. So as a result, thanks to him, we have many tryptamine derivatives and LSD derivatives and phenethylamine derivatives and all of that. Their activity as psychedelics is confirmed through animal studies, through receptor binding studies, and occasionally through what you might call the large animal bioassay, which is kind of the confirmatory assay that you need to do. And so, that gives us an even broader palette of, if you want to call them molecular probes or chemical tools to investigate these processes. So when we talk about the true psychedelics, we can talk about the materia medica of this. And the four biggies are LSD, psilocin, and mescaline and DMT, dimethyltryptamine. Note that two of the four, psilocin and DMT, resemble serotonin quite closely. There's really trivial differences, but critical differences. LSD is a little harder to dissect out the tryptamine nucleus, but it too, really, you can think of it as a complex tryptamine. Mescaline is the odd one out. It's a phenethylamine, but it's representative of also a class of true psychedelics. DMT is quite widespread in nature. It's found in many plants, many animals. We'll talk more about that in the second talk. Psilocin and psilocybin. Psilocin is the active form of psilocybin. Psilocybin is converted to psilocin in the body. But psilocin and psilocybin are quite widespread in the mushrooms. There are about 200 species of mushrooms that contain these things. And so far, nowhere else, no animals, no plants contain these. They are fungal products. Mescaline is not so widespread in nature. It's found in the cactus family and quite a number of cacti, and that's basically it. Not found so far outside the cacti except for one anomaly, which I won't get into here. But then we also have the quasi-psychedelics, and if we're talking about the quasi-psychedelics that are strictly speaking not 1A or 2A agonists, but still quite useful for the study of altered states of consciousness. One of them is salvinorin A. It is not an alkaloid, so it lacks nitrogen, unlike most of the psychedelics. It is not a serotonergic drug. It works on the kappa opiate receptor. It's quite selective and quite potent for the kappa opiate receptor. So this work is important because it's opened up a whole new area of psychopharmacology that we haven't looked at that closely yet. An incredibly useful tool for studying it because of its selectivity and its potency. Brian Roth at the University of North Carolina in Chapel Hill is the person who really elucidated the pharmacology of salvinorin A and demonstrated that it was a kappa opiate receptor. We had an interesting conversation in the sense that he said, had I set out to design a highly selective kappa opiate receptor ligand using computer molecular modeling and so on, it wouldn't have been this one. This one doesn't look like any of the theoretical models. It's a lipid-like compound. It's quite nonpolar, and it doesn't, you never would suspect. So as an ethnopharmacologist, that was very gratifying because what it tells me is that, ethnopharmacology, which has the benefit of thousands of years of human experimentation with plants, trumps computer-aided molecular design every time because it has the benefit, well, not every time, okay, not every time, but it has the benefit of that human element of experimentation. Another really important one that we're all familiar with is a synthetic compound, MDMA, methylene dioxymethamphetamine. And it works on serotonin, but it doesn't work on the 1A, 2A receptors. It actually works on the so-called serotonin uptake site. And if Prozac is an uptake inhibitor, you can think of Prozac as a monkey wrench that kind of jams the uptake inhibitor so that serotonin can't be taken back up into the presynaptic neuron. MDMA jams the valve open so that all of the serotonin leaks out in one sort of massive spasm, and that's why it depletes serotonin over time. But it has a different effect. But it is a serotonergic drug. It just doesn't work the way true psychedelics do. So we've got those tools. Now psilocybin emerges as quite interesting in this respect because they're so close to the neurotransmitter serotonin. And as a result, they are quite compatible with human metabolism, human and mammalian, I should say. We are well equipped with the enzymes and the molecular machinery we need to metabolize psilocybin. They're orally active. They don't require any monoamine oxidase inhibitor to become active. You just eat the mushrooms, no processing necessary. And they work. So they're very compatible with our own neurochemistry. And as a result, they're quite suitable for therapeutic use, especially in patients who might be in a fairly fragile state of health, such as cancer patients and this sort of thing. They don't cause catharsis, they don't cause purging like ayahuasca and that sort of thing, and that may have its place too in the therapeutic spectrum, but psilocybin is gentle and short-acting and unthreatening, usually, largely, phenomenologically, depending, of course, on a lot of things, the classic set and setting. But it's a safe drug to use in the clinic, and it can be used with people who may have health problems or maybe are not, as adapted to these things. So it's a real good drug. It's a good study drug as well, and I think it's probably got the best chance of actually getting into the clinic as something that is used and approved by the FDA for regular use in certain kinds of therapies. Now, one of the applications of these psychedelics, and I thank Alex Gray if he's in the room, I've shamelessly ripped off his one of his paintings. I'm sure that I'm the only person that ever did that, but thank you. He depicts it so well, you have to expect that the guy knows a few things about this pharmacology, this phenomenology. But one of the applications of these compounds is they're excellent tools for investigating this neural substrate of consciousness that we're all so fascinated with. It has a well-characterized pharmacology, a 1A, 2A agonist. It's a classic real psychedelic, true psychedelic. And it's being used a lot, both in basic neuroscience and also in the clinic, in the clinical applications. I'm going to talk about some of that now and at some level of detail, which must necessarily be superficial. So I'm going to talk about the Hefter researchers first. David Nichols, Franz Vollenweider, who is here at the Zurich Hefter Center. Dave Nichols is a professor of medicinal chemistry and pharmacology at Purdue University. Charlie Grobe, many of you know him. He's a clinician and at the moment the principal investigator on our psilocybin cancer study. And Mark Geyer, who some of you may know if you're in the field, and some of you may have never heard of. He remains in the shadows by choice, and he and Franz work quite closely, and a lot of what he does is involved with animal models, but all stellar researchers who do not need really to be recognized in the psychedelic community to be recognized as very, very good researchers, and they've all had grants for years. And so Dave focuses on, as I mentioned, the structure-activity relationships of hallucinogens, and some of you may know about this from Sasha Shulgin's work. He was really the pioneer of this. You can take a molecule and do theme and variations. You can tweak it slightly and then test it and see how is that different from the parent molecule. And that all has to do with how these things bind to the different relevant receptors. So he invents these tools and then he uses them to characterize these receptors. And the benefit, the side effect is that he also can make these things available. Well, he publishes. He doesn't make the compounds available, but he makes the information available. And he is concerned with the trafficking of so-called second messengers, because when a drug binds to a receptor, there's a whole lot of things that go on, or not. But one of the things that go on is the activation of second messengers, intracellular messengers that may lead to gene expression modulation and this kind of thing. He's interested now in looking at the non-serotonin mediated pharmacological actions of LSD. One of the anomalies of LSD is that it's an order of magnitude more potent than any of the other true psychedelics. Why should that be? We're not sure. He's not sure. But it seems that there are signaling pathways that are responsive to nanomolar and subnanomolar, possibly picomolar levels, doses. And so that's a whole sort of area that he's gotten interested in the last few years. He's also involved in dopamine, which is another major neurotransmitter, and it's involved in schizophrenia and also Parkinson's disease, and he does a lot of dopamine research that he publishes extensively on, and we don't pay much attention to, we psychedelic people, but very interesting. And he's got, some drugs that will probably be major treatments for schizophrenia that will be coming out in the next 5 years, God willing, 10 years, whatever. He's done a lot of work with MDMA, both behaviorally, using animal models, and also looking at the molecular pharmacology. He's invented many analogs of MDMA. And he did our whole community a great service by founding the Hefter Institute in, I think it was 1991. And he is a great guy. If any of you know him, we all love him. And he brings a certain gravitas, I guess you could say, to our work, as well as a good sense of humor, and he's a fine man. We're very lucky to be associated with him. Now here in Switzerland, we've got Dr. Vollenweider, Franz Vollenweider, who's the director of the Hefter Research Center at the University Hospital in Zurich. And he's the fellow that I mentioned in my talk. We visited him yesterday. And he is in a better position because he's able to work with human subjects. He's gone through all the regulatory hurdles, and he's able to use his rather impressive tools, instrumentation, and psilocybin and other compounds to look at the basic neuroscience of consciousness and brain dynamics. So he uses PET imaging, positron imaging, to look at the 5-HT2A receptors. He uses a three-dimensional EEG to compare psilocybin and meditation states of consciousness. He uses 5-HT2A schizophrenia-like psychosis induced by psilocybin, effects of psilocybin, altered states on cognitive functions like neuronal switching and binocular rivalry. I'll talk a little bit about that, their effects on the perception of time, our oscillatory processes that regulate our temporal subjective sense of time, its effects on working memory, its effects on rate and rhythmicity of perceptual rivalry. That's the competition between hemispheres in perception. And all of it, as you can see. And clinically, I mean, all of these are clinical studies, but they're not therapeutic. On the therapeutic level, we're interested in the effects of psilocybin on bulimia and other eating disorders, because these disorders are in some ways a dysfunctional state of body image. And we know that serotonin is involved in that and is also involved in eating behavior, craving, and that sort of thing. So we're in the process, Franz, I should say, of developing a clinical protocol to look at anorexia nervosa and bulimia, and we expect to get that approved. So one of the most spectacular and at least pretty things that he does is he uses positron imaging technology to show what parts of the brain light up when you take psilocybin. And this is just a comparison of the brain at its resting state. The red areas here are the areas of highest metabolic activity. The ligand here is a form of glucose that, as it's metabolized, emits positrons that can be detected by the instrumentation. So you can show what this shows essentially is where is the action going on in the brain, and you can sort of use computers to cut this section or slice out and look inside while it's actually happening. This is a static image, but you can also make movies that show what's going on. So this is a very powerful technique if you want to localize what goes on in the brain when psilocybin or psilocin, its active form, binds to its receptors and does its thing. Another thing he's looked at that's quite interesting that I have a hard time explaining to you because I'm not sure I completely understand it myself, but is switching. Switching is exemplified by this classic notion here, where in this image you see either two faces or you see a vase, or a vase, as we would say in Colorado. And so, and there's really no way to control. Sometimes you see it like a vase, and sometimes you see it like faces, and you really don't have much volition over that. So he has developed an interesting method with this simple computer-generated graphic where the sphere is either rotating one way or it's rotating another way. And if you look at it for a while, you can see how it switches. And he's looking at what are the effects of psilocybin on this as opposed to normal consciousness. And why is that important? Well, that's where my understanding falls down, except that it shows, there's the sort of competition between these different perceptual modalities. This is basic stuff. I mean, it seems trivial, but you have to get trivial. You have to understand the basis of perception before you can sort of build on that foundation. So he's doing that work. One of the interesting other things, many interesting things he's doing, is this so-called binocular rivalry. Dr. Vollenweider just emailed me these slides. They weren't in the original presentation, but I felt that I ought to try to get them in because he went to the trouble of it. But the basic setup of this protocol is they present vertical and horizontal gratings to a person wearing a pair of glasses alternately to each eye, to the left eye or the right eye. And every time the gratings are presented to the right eye, the left eye is blocked and vice versa. And the task of the subject is to click a mouse every time that happens, which sends a signal to a detector. And what he has done, I guess I should have run the animation a little earlier. What he's shown is that the frequency of this switching, this binocular switching, can be affected or modulated by normal perception. And in normal perception, we have a certain rate that this goes on all the time, and it's kind of stretched out over different time periods and different frequency amplitudes. And psilocybin will change this, not surprisingly, and other things like meditation will change it. So one of the things you can do is compare these two modalities of altered states. You can look at psilocybin, and you can look at meditation. And when you do that, you find that there's a similarity. Actually, as time goes on, they begin to resemble each other. And at moderate doses of psilocybin, as you settle down, the frequencies become dampened down, and the time frequency becomes much more concentrated. And in meditation, after the person has gone into the state for a while, you see a very similar reaction, a very similar kind of response. So this shows that there's fundamentally a homology, if you will, between deep meditative states and deep psilocybin states. So there's that. Okay. That's all I'm going to say about it, because that's probably about all I can tell you about it. Now, on the clinical side, we've got Dr. Grobe at the Harbor UCLA Medical Center, and his work is not so visually striking because it's clinical. But he was the clinical. Somebody telling me, oh yeah. He was the clinical investigator on the ayahuasca project, which is how I got to know him. This study we did in 1993 with the UDV in Brazil. And he has also done subsequent work on ayahuasca, working with UDV investigators on a study of the adolescent use of ayahuasca, or huasca as they characterize it in Portuguese, in adolescent UDV members to sort of get some parameters on how they integrate into society, and are they screwed up by this and so on. And in fact, quite the contrary. They're very, well-adjusted kids by and large. He has run a psychobiology of MDMA study approved by the FDA, basically a kind of a basic neuroscience study to try to characterize, the psychobiology of MDMA in human subjects. He's now the principal investigator on this psilocybin cancer anxiety study, where the psilocybin is used in terminal cancer patients to help them deal with the anxiety of their approaching death, help them relieve pain and sort of come to terms with it, obviously in conjunction with intense therapy as well, but kind of loosening up their their fears and helping them to really, in a sense, die a good death. And so that is going on right now. And the idea is well, I think I got ahead of myself here. The idea is to determine if treatment with psilocybin can help reduce their anxiety, their pain, the depression associated with that. And that's important because pain and depression are both treated with other types of medications. So if you can reduce pain without using narcotics, the person can die more clear, can retain clarity when they go through the dying process. And that's good for them and good for their families usually. He initially slated 12 subjects for the study. 11 have completed this protocol, and now the FDA has allowed him to expand one more patient. So he now gets to add 2 additional patients. Excuse me? Oh, okay, there's echoes, or I'm hearing things, I don't know. The results, the outcome of this has been very positive so far. In fact, so positive in the case of one of our patients that the husband of the woman that underwent the therapy was so moved and touched by the outcome that he volunteered to become our fundraiser, our member of director of development, and that. His name is Norbert Linsinger. I was hoping he would be here, but he wasn't able to make it. But so it's been very gratifying because this is one of the first demonstrations that it's effective, it's safe, and ultimately the Hefter mission is, as we stated, is to alleviate human suffering. In this case, we are fulfilling this mission. Psilocybin is a good medicine that can be used in this way. One of the basic clinical protocol is initial psychotherapy. It's a classic double-blind placebo-controlled methodology. The placebo is niacin, which causes a kind of flushing but not much psychological effect, and psilocybin is the active, 0.2 milligram per kilogram. It's a general clinical research center setting, which isn't the best setting to take something like psilocybin in, and it's a pretty sterile situation if any of you have ever seen these things. And then, of course, there's evaluation and follow-up of the results here. Certainly one of the benefits that's come out of Charlie's study is the decor of the clinical research center has decidedly improved. This is the before and after picture of the clinical unit where this work is done. Hang a few curtains and make it nice, so that people can feel more comfortable when they have these experiences. Whether they're going to implement this all through the clinical research center, I have my doubts. But they did make that sort of concession to the issues of set and setting. You can look at these websites, canceranxietystudy.org or clinicaltrials.gov. You'll, I think, go to the same website in either case. Charlie is still looking for 2 patients. It says 1 here, but then we got the approval just a few days ago to add another one. If any of you know a patient or a person that might be a good candidate, give him a shout, get in touch. You'd be surprised how difficult it is to recruit patients for a study like this. You'd think people would be lining up at the door, but it's not the case because the inclusion criteria are strict. The person must be dying of cancer, but otherwise healthy. It's a difficult, standard to meet. So please help. If you really do know somebody, please help. This is just a little thing I threw in about the alternate reality salad, made from mushrooms with the chef's own garden, from the chef's own garden. Mark Geyer is the UCLA professor of psychiatry and neurosciences, And his work involves the neurobiological effects of drugs acting by these, again, monoamine neurotransmitters such as serotonin and dopamine. He uses animal models a lot. He doesn't do too much with clinical studies as yet. His real focus of research is the physiology and neurobiology and pharmacotherapy of schizophrenia. And there are quite there are similarities between psychedelic states and schizophrenic states. It's not all just mysticism and happy, happy, joy, joy. And so he looks at schizophrenia in animal models and preclinical models. And he has a powerful tool himself that he uses extensively, the so-called gene knockout mice, where you can actually breed mice which don't have receptors for these key neurotransmitters such as glutamate, dopamine, and serotonin. The mice live, and whether they're happy is, I don't know. Nobody asked the mice, but it's a very powerful technique given these methods of looking at it behaviorally and these behavioral protocols. For example, he talks about habituation and gating. Under normal conditions, you have sensory and cognitive information coming in, and it passes through various filters. And then after whatever gets through the filters is processed, that's further integrated and processed until finally you get a coherent schema that we experience as reality. This is the consensus hallucination that we call reality. And in altered states, the same thing is going on, but the filters are disrupted. They're deficient in some respects. So you get a lot of information pouring in and no real way to exclude it. This is the idea, Huxley's idea of the doors of perception, that, you know, senses are as much a reducing mechanism as they are a reception mechanism. And so you get information overload, and the poor schizophrenic or the lucky psychedelic voyager is suddenly overwhelmed with information, and depending on what they do with that, they either find it a novel experiential process or a rather terrifying one. And so he uses that kind of methodology. One of the examples of this is habituation. Is one of the simplest forms of learning. You can animals or humans, you can use what's called an acoustic startle effect, which is essentially, you know, rapping, making a sharp noise, and a person in a normal state will eventually accommodate to that and not really be affected, not really be startled. And so that's a habituation. And in a schizophrenic, it doesn't work that way. They don't habituate quite as readily. So they remain, even though the stimulus is repeated many times, they remain sensitive to it, and that disrupts perception and experience. So that's kind of what the Hefter is doing. We also, through fundraising and collaboration, and we help with the design of clinical protocols. We review the protocols of other investigators. We, along with MAPS, as you know, we try to get money to support other types of studies. And a couple of the ones that have gotten a lot of attention in the mainstream media and also in the scientific field is Dr. Roland Griffiths at Johns Hopkins and Dr. Francisco Marino at University of Arizona in the Department of Psychology. And Dr. Griffiths' work is very interesting because he's actually been able to get approval to investigate the use of psilocybin in the elicitation of mystical states and religious experience. And you may be familiar with the Good Friday experiment done these 40 years ago. I'm not sure, I think about 1964, but it's appropriate that today is Good Friday, where theologians were given psilocybin in a double-blind experiment in a church during a church service, and it was they tried to determine if they were really having a mystical experience or not, or how mystical was their experience during this service. And there were difficulties with the design of the placebo blind. Many subjects that received the active dose were kind of rolling on the floor laughing, and some of the ones that received the placebo were not so stimulated. So it's kind of, but we've come a long way since then. And Dr. Griffith is to be credited for designing a rigorous double-blind placebo-controlled study to look at these mystical states, these states of religious ecstasy, essentially. The drugs that, psilocybin again was the drug of choice here, but other drugs, LSD and DMT, are also good pharmacological models for these mystical experiences. So now we actually have a tool to understand or study transcendence using these substances as pharmacological probes. So what are the brain states associated with transcendent experience or mystical experience? This may be criticized by some as reductionism, but we're not really going to debate that here. This is it's a tool that we can use. And he sought to answer this question initially posed through the Good Friday experiment. Can psychedelics in an appropriate setting trigger or stimulate mystical or religious experiences? He found quite striking results. Of 24 subjects that received psilocybin under these strictly controlled conditions, 15% rated it as the single most meaningful experience of their lives, while 60% rated it as among the top 5 most meaningful experiences of their lives. And after 6 months follow-up and 1 year follow-up, that pretty much held. And interviews with the family members and other loved ones of these people also supported this, in the sense that, John Smith or Joe Blow was a real bastard before he took psilocybin, and he's so much nicer now, So it helps, and it's important, to be able to study this. I mean, if we have a tool that can reliably induce mystical experiences, it can help us in our spiritual evolution. Is time? Okay, well, all right, well, we're getting there. I have 2 hours, and the next one won't be so hard. I'll try to speed up a little bit. Obsessive compulsive disorder, as we know, is a psychiatric disease that is characterized by recurring obsessive behaviors, repetitive behaviors such as hand washing and that sort of thing. It's the most commonly diagnosed, 4th most commonly diagnosed psychiatric disorder. Because it's very hard to control the behavior, it is also associated with depression and substance abuse, and interpreted as an effort at self-medication for the problem, suicidality, panic, interpersonal difficulties. So it's a problem. And anecdotally, reports emerged that people who were abusing psilocybin or LSD or some of these things sometimes had dramatic remissions from their OCD symptoms that lasted for months beyond the trip itself, and sometimes permanently went into remission. So Dr. Marino at the University of Arizona said that's interesting, and obtained the FDA-approved study for psilocybin to treat OCD. And initially, he's only done 9 patients, and the initial study is a safety and tolerability study. Most patients experienced a reduction in these symptoms. So it's looking good. It's far from definitive, but it's very promising. So now, as things go in science, the next thing is to do a larger study. So what's next? Well, there's a great deal of interest right now in ayahuasca. Ayahuasca is the drug of the moment, perhaps. And it's got its own problems in terms of studying it clinically, but ayahuasca is traditionally used in shamanism, of course, but it's also being used to treat addictions in South America and other places, especially alcoholism, but they're not really controlled experiments. These folks are just applying it clinically, and they're not really trying to assess statistically or any of the effects, but somebody needs to do this. So one of the interesting outcomes of our UDV study with the Brazilian group was that we were invited to participate in this study. It was an international collaboration with Dr. Grobe as the principal investigator. And this has come to be known as the Ayahuasca study, which you probably read about in the MAPS bulletin and other places. So of our initial findings, it was an observational study. We weren't giving the substance to the subjects. They take it as part of their ritual, as part of their sacrament. So they were kind enough to let us come in and attach EEGs and take blood samples and this kind of stuff. We found that there was no acute toxicity associated with ayahuasca. There was no evidence of neurological deficits or cognitive impairment or personality disorders in users that have used it anywhere from 10 years to 40 years. That was the time span that our studies covered. Most of the members that we looked at had histories of drug abuse, alcoholism, and domestic violence before they joined the UDV and before they started consuming ayahuasca regularly. Most of them told the story that they had initially very terrifying experiences which motivated them to change their lifestyle, change their behavior. And they also universally credited the UDV as the vessel, the setting, if you will, and also the set, to a certain degree to enable them to maintain this changed behavior after their ayahuasca experiences. So they became good citizens and good fathers and parents and good husbands and wives, and their lives improved. And one of the interesting outcomes of this study is we found there was an unexpected upregulation, an increase in the abundance of the serotonin transporters in these subjects. This was after a period that was allowed to allow the ayahuasca alkaloids to wash out of their system. Then we measured this in platelets, and we found, these serotonin transporters are elevated. They're more dense, so that means the transporters are more efficient transporters of serotonin. What does that mean? We didn't really know. It didn't really change the affinity of the transporters for serotonin, that being the other measure. In other words, how much it, in a sense, likes the transporter at what concentrations. But the upregulation in the abundance or the density of the transporters was interesting. So then we wondered, what does that mean? And we looked into the literature, and we found, oddly enough, that deficits in these transporters have been shown to be associated with certain types of alcoholism. I was informed by Steve Pallas today, type 2 alcoholism, alcoholism associated with violence and tendencies to suicide. Suicidal behaviors has been associated with this. Binge eating behaviors have been associated with these deficits. So it appears that ayahuasca may actually be able to reverse these deficits, may actually be able to cure on the neurochemical level what we see change for the better on the behavioral level. So we're interested in getting an ayahuasca study going. And hopefully this will be the year that we can get protocols, if not approved, at least on the path toward approval, and look at this phenomenon. It's very promising. Ayahuasca in traditional medicine is certainly a healing medicine, and we'd like to bring it into the clinic if possible. So, that's the end of the first talk.